Hello, everybody. Thank you very much to the organisers for the invitation. Thank you very much to Lars in particular. I've been a fan of this conference over the years. I've always watched the videos online after on YouTube, so it really is an honour to be able to present this year, although a shame not in person. The virtual format is still nice. And it's really nice to be following Lars as well in what looks like a really interesting symposium. I'll be picking up some of the concept I'm sure Lars has introduced around human milk oligosaccharides, and building upon that with the importance of the microbiome in preterm infants. Some disclosures and disclaimers. And I think this audience particularly probably have a very good handle on what the microbiome is, quite literally the small community that occupies a distinct environment. For the purposes of today's talk, that will very much focus on the bacteria in the gut. But it's important just to be aware of other microorganisms that colonize the gut. So you have archaea and fungi, bacteriophage, that are viruses that infect bacterial cells, and also human viruses. And in adults, at least, around 56% of the cells in the human body come from our microbes, and less than half from our own human cells. When we consider that at the genetic level, less than 1% of all genes in the human body come from our human genes, and over 99% come from our microbes. And I think over the course of human history, we've had a very good understanding of the role of microbes in disease. And I think what we're increasingly appreciating is the role of microbes and bacteria, in particular in human health. I won't go through all of these, but just to note a few that I think are particularly relevant for early life: resistance against pathogens or pathobionts. Providing that protection against epithelial injury is crucial for diseases like necrotizing endocolitis, which I'll come on to talk about. And also something I won't talk about today, but I think is very important and will over the coming years, I think, be crucial in our understanding of complex diseases, is how the microbiome and the immune system interact during especially the first year of life, which is likely the key time frame for human development. And again, just to introduce the topic and what will be relevant when I'm talking about some of our recent preterm data, I wanted to introduce some of our previous work around term infants. And so here, this was a large cohort called TEDDY, around 12,500 stool samples sequenced from around 1,000 infants across 6 different geographical locations. And the study itself was focused on diabetes, but with such a rich data set that it could also be used to just define what factors are associated with microbiome development in early life. And that's exactly what I'm showing here. First of all, on the left, we're looking at the bacterial species. You can see our first time point of month 3 to month 6, and you see quite clearly breast milk is the main factor associated with microbiome development at that time period, and other factors, for instance, birth mode. If we fast forward to 1 year of life, again, you see breast milk is still the most important and most significant factor associated with microbiome development. Birth mode, we also still see, and other more environmental factors also significantly impacting microbiome development. After 1 year of life, we really no longer see much breast milk in the diet, and we no longer see that as being significantly associated with microbiome development. And indeed, by month 19 of life, each individual has become very individualized in terms of their microbiome, very stable, and is not really perturbed by any of the other covariates anymore. And that applies for the remainder of the sampling. So that was looking at bacterial species. When we look at bacterial function or functional capacity, we see that only breast milk was associated with the microbiome functional capacity, and again, only over the first year of life, after which breastfeeding really seldom occurred, and we no longer see that as being significantly associated. Again, this is published. I won't go into any further details on this paper other than this slide. And I guess behind the scenes of this slide is some quite complex modeling, which you can feel free to go and dive into the extended data within this manuscript. It's all open access. But just for visualization's sake, I think this is a really nice plot just to show us breast milk declines from the diet over the first year and a half of life. You see those corresponding decreases in several of these different bifidobacterium, particularly Bifidobacterium breve. And this hopefully follows on nicely from what Lars was talking about in terms of human milk oligosaccharides. We know there's several functions for HMOs, and I'm sure there's many more to be discovered. but one of the primary roles is to serve as a prebiotic, particularly for bifidobacterium. And so perhaps it's not a surprise here that as breast milk and HMOs are reduced in the diet, we see corresponding reduce or decline in some of these different bifidobacterium species. There was further work done in a sister paper published by Tommy Vatanen, who's now in Auckland, New Zealand, looking at the HMO utilization genes of these different bifidobacterium. Again, I won't present that data today, but it's worth having a look at if you're interested. And in essence, we observed that whilst breast milk was in the diet, there was much more abundance of genes responsible for the utilization of HMOs. And when breast milk was removed from the diet, those HMO utilizing genes generally diminished from the infant microbiome over time. So really that sets us up for our preterm work, which is now the primary focus of my lab here in Newcastle. I'm very lucky to work alongside clinical colleagues, particularly Dr. Janet Berrington and Professor Nick Embleton, who really have worked tremendously hard over the last 10 years to build a large research basis here in the northeast of England. And from infants born significantly premature, so less than 32 weeks at gestation, less than 1,500 grams at birth, they've collected a huge range of sample types, all of which I haven't listed here, but the main samples that I use for my research are described here. And you can see the various applications that we use these samples for. For instance, we collect maternal breast milk, and we, in collaboration with Lars Bode, profile HMOs. We also look at microbiome in breast milk as well as stool. And stool and urine, we also profile functional small molecules by volatile organic compound analysis as well as metabolomic analysis. And in terms of more invasive samples, such as salvaging blood and resected tissue, we have really good protocols in place that allow us to collect these samples that would otherwise be discarded. So not for research purposes primarily, but whatever is otherwise considered waste can come to us in the lab. We can use those samples to directly perform, for instance, microbiome analysis at the site of disease, immune profiling, and most recently to generate human intestinal derived organoids. And I'll mention a little bit about that work at the end of the talk today. All of these samples are housed in the Great North Neonatal Biobank here in Newcastle, some 100,000 samples from over 1,000 babies in that resource currently. It's available not just to myself locally, but also investigators around the world. So if you are interested, please reach out to us. We're always happy to help and support and collaborate with other research. And equally, we've got quite a lot of data now generated from these types of samples. If you're interested in accessing the data, very happy to share that. So here's an unpublished study, largely performed by my newest recruit, a PhD student, Lauren Beck. It's of healthy preterm infants only. All the data was generated using metagenomic shotgun sequencing. And here we have 124 preterm infants and nearly 1,500 stool samples. So a reasonably large cohort, certainly the largest healthy cohort with metagenome data to date. Much like the earlier study I mentioned around diabetes, we were very keen to define what covariates were associated with microbiome development in this exclusively premature population. And much to our surprise, factors like gestational age and birth weight were not significantly associated with the overall microbiome profiles. Delivery mode also wasn't, although we have published previously to show that actually we don't see delivery mode as being a main driver of microbiome in preterms, unlike, of course, in term infants where you do see that. This may well reflect the universal antibiotic use on the unit following birth. And I think so it's notable some of these absences, but also notable some of the associations that we did find. So antibiotic, breast milk, and breast milk fortifier all at some point were significantly associated with the microbiome profiles. And perhaps to nobody's surprise, probiotics were also significantly associated with an altered microbiome development at all time points except the first here. And this probably reflects that probiotics were used from day 8 on average. And they were stopped around day 44 on average, or on a median of day 44. So we do still see some effects afterwards, and I'll talk about that in the coming slides. Just to give you a little background onto some of the techniques that we use, which in and of themselves don't really tell us anything, but we can apply them to models and transition network plots that would otherwise not be possible to perform. These are dimensionality reduction techniques. So we use Dirichlet multinomial modeling or DMM clustering. And this allows us to take all the 1,500 stool sample metagenome data that we have and distill it down into a defined number of groups. In this case, the algorithm determined 5 groups to be optimal. We then number those groups, 1 being the group that contains, on average, the youngest samples, and group 5 being that contains, on average, the oldest samples. You can see via this heat map the type of distribution of these different species that we have, largely confirmed here by our Lefse analyses. And really, just to give you a sort of very high-level view of this, you see that Staphylococcus generally dominates cluster 1. Preterm gut community type 1 is what the PGCT stands for. Escherichia, both E. coli and unclassified, dominate preterm gut community type 2. Klebsiellas in number 3. Bifido-Anomalis and longum, particularly in cluster 4. And again, here you can see the Bifidobacterium breve dominate in these cluster 5. And again, that's reflected here in that heat map. So as I said, the real purpose of that is to allow us to do techniques and analyses that we would otherwise not be able to, the main one being this transition network type analysis. And so here's just a reminder of those dominant taxa across those different clusters 1 through to 5. And you can see here are different time windows. And this population in this particular view had no probiotics. So going back 10 years, we didn't use probiotics on the unit, and these were collected over the period of 2010 to 2013. And you see that most infants start in cluster 1, which is our Staphylococcus rich group, and that's quite common. And they generally transition towards clusters 2 and 3, so the Escherichia or the Klebsiella group, and they tend to remain quite stable within one of those 2 clusters once they transition there. In around 2013, middle of 2013, the unit introduced Influran probiotic, which contains B. bifidum and Lactobacillus acidophilus. And you see a very stark contrast to the microbiome transition in that population. So here, even within the first time point, you see some infants already have transitioned into preterm gut community type 5. Certainly throughout their sampling, they're much more likely to transition and remain within this preterm gut community type 5, which is our breve dominated cluster, which is interesting because breve is not part of Influran. And finally, after I think around 2017, we introduced Labinic or LB2 into the unit. And again, you see a very different profile from that to no probiotics, but also that to Influran influence. So although the probiotics are very similar, only difference being Labinic has an addition of Bifidobacterium longum subspecies infantis, you see that in those infants, they generally transition towards cluster 4, which is our B. longum, B. bifidum group. And it's worth noting, as I said earlier, we deliberately call these clusters 1 to 5 based on their average age of samples. So by proxy, clusters 4 and 5 could be considered our most mature. And so the 2 take-homes here are, I suppose, Influran and Labinic certainly increased transition into our most mature gut microbiome community types, preterm gut community types, and also, regardless, they are both increasing the amount of bifidobacterium in these infant guts. Here you can again just see the very stark contrast between the 2 different probiotics. So our never group is in the middle, and you can see, depending on which probiotic they get, they sort of head off or drive in very different directions based on their microbiome profiles. So again, just underscoring the importance of probiotic selection, even when the same species are contained largely, clearly they're different strains and are having here very different impacts on the infant gut microbiome. So just to look at the prevalence, so less focused on relative abundance, but just whether or not we could detect those probiotic organisms. For B. bifidum, which was present in both, we were able to detect that before probiotics were given. So again, suggesting at least DNA from these probiotics is detectable in the stool before the infants ever received that probiotic. We detected during and after, which is less surprising, and much higher prevalence compared to our never group. You might wonder how we have a before, during, and after never population. That's simply just age matched based on the average ages of these different windows. And again, you see differences between the strains. So the Infloran B bifidum has a much higher prevalence compared to Labinic, but of course, both are significantly higher than the never. Here we have B longum, so that's only present in LB2 or Labinic. Again, we see the before colonization, as expected, again, during and after, a very high prevalence. But interestingly, we also pick this up in Infloran as being significantly higher in our probiotic versus never group. And here again, lending support for this bifidogenic effect, where the presence of some bifidobacterium, in this case B bifidum, is perhaps supporting the growth of other bifidobacterium, for instance, longum. And finally, Lactobacillus acidophilus, which was in both probiotics, is higher than the never group at all these time points. Again, we're detecting it before. And I think it's always striking for Lactobacillus acidophilus, especially how sort of rare it naturally occurs in preterm infant populations, yet is used quite extensively throughout different probiotics. Okay, so now I'll talk briefly about some of our published work. And again, a disease that probably needs no introduction to this audience is necrotizing endocolitis, the leading cause of death in preterm infants, and is basically an exaggerated inflammatory response leading to necrosis of the bowel, most commonly the terminal ileum. And in the worst cases, those infants will undergo surgery to have those regions resected. So we know that human milk oligosaccharides and the microbiome have been associated with this disease through our work, through Lars's work, and many others around the world. And so this has been an area of great interest for us over the last decade or so. Just to pick up on an animation from a paper published a few years ago now, here we have 6 preterm gut community types because we're dealing with 16S data rather than metagenomic data. Don't ask me why it clusters 6 ways with 16S and 5 ways with the other, but it does, and that's pretty consistent in our hands. Here, each dot represents a different stool sample, and it's colored based on which cluster that infant sample derived. So basically, the yellow here is our bifidobacterium dominant cluster. You'll see during the animation, our neck infant and control infant both start in the Staphylococcus rich group. The neck infant shows much more chaotic or less stable development over the initial weeks of life, contrasting that with our control infant who moves gradually from the Staphylococcus rich group into the bifidobacterium rich group and then remains in there for the remainder of their sampling. So this is 2 arbitrary cases, but I think it makes the point quite well. We applied statistics to this in the published manuscript, again, open access, and showed that infants who went on to develop neck had significantly less stability and significantly less relative abundance of bifidobacterium. So we followed this up in collaboration with Lars to look at maternal human milk oligosaccharide profiles, showing that neck and control infants have significantly different HMO profiles, or infants who go on to develop neck versus those who remain healthy controls have received significantly different HMO profiles from mother's milk. Very nicely, and this very rarely happens in science, when we looked further into see what HMO was driving this difference, it was the same HMO that Lars had shown in his earlier work, being diacylylacto N-heterose, was really driving that difference between neck and control. So a really rare moment in science where you get really good corroboration of previous work from a completely different area, different numbers in a different population. We applied some univariate models to that, determining 241 nanomoles per mil would be the optimal threshold in our cohort. This would require validation and more cohorts to begin to make that more generalizable. But within our NICU, at least, we would have very good sensitivity and specificity using a threshold like this. And you can see here basically how our control infants generally almost always above that, and our neck infants generally below that. And finally, using that clustering approach, we also were able to show that infants who had above the DSLNT threshold generally progressed into those clusters 4 and 5, which is our more mature microbiome profiles dominated by bifido, and infants below that threshold generally did not transition towards clusters 4 and 5 and remained in the more staph and other pathobiont-rich clusters. Okay, I promised I would briefly mention the organoid work that we have ongoing. For those unfamiliar, when we get tissue samples from our preterms, we isolate the crypts, generate these 3D enteroids. We then differentiate them into all the major cell types of the gut. And we have this model whereby we can keep the bacteria in anaerobic conditions, the human cells in oxygenated conditions, and then model how they interact over a period of time. And we've shown very nicely using preterm versus adult enteroids or organoids that there's a clear difference. And what I thought was really interesting here is when we add the microbiome, that's when we see the biggest change in expression and gene expression between preterm and adult derived organoids. So lending some evidence towards this being a useful model for studying premature disease such as NEC. I won't go through the summary. If you're watching this as a recording, so you can press pause at this point. I would like to thank the various members who's been involved in this work, particularly Andrea and Lauren, to Janet and Nick, without whom none of this would be possible, and to Lars for his support with the human milk oligosaccharides, to our funders, particularly the charities and with whom we work very closely. And with that, here's any contact information, and I would be delighted to answer any questions you might have. Thank you for your attention.